Hello, everyone. I'm Bastien, and this is James from the Geo Lab and the Portela Research Group in MIT Mickey. And today we're very excited to say a few words about our image and how we designed fully tunable 3D microscale cell scaffolds. Tumor cells and others have to migrate through mechanically diverse environments during metastasis, from stiff to soft, from porous to confined, from stable to dynamic, and so on. In particular, some of the most aggressive cancers can migrate through various tissues with ease. However, current methods to study cancer cell migration in lab settings are somewhat limited because the materials used, typically soft hydrogels, can only mimic a narrow range of conditions. One may be able to tune the stiffness of their gel by changing its cross-linking density or the condition of its polymerization, but options are limited, and tuning mechanical properties independently is generally not possible. What is more, the resulting medium is almost systematically homogeneous, very far from the mechanically diverse physiological conditions. In this study, we decided to explore how we can leverage microscale 3D printing to generate fully tunable cell microenvironments. As you can see in this close-up, we are able to engineer mechanical properties locally, at a cellular scale, and in a non-coupled fashion. For example, this means that we can choose to have an environment to be stiff or soft while maintaining its porosity. We envision that this platform will allow us to study how certain cell types change their behavior or phenotype in reaction to local changes to stiffness, porosity, and so on.
All right, so how do we make these architectures? So we use a 3D printing technique called two-photon polymerization, which is not exactly a common technique, so it's okay if you have not heard about it. Basically, it involves shining a focused laser beam onto a photoresist, and the spot at which that beam is focused on will turn that photoresist from liquid to solid. That spot is then moved around in 3D space to create the 3D architecture. And this technique has allowed us to print features in the order of 1 to 2 microns. And based on this technique, we can create a diverse range of 3D architectures with varying mechanical properties. For example, we can make structures which are highly stiff with straight monolithic beams, or highly compliant and stretchable structures with woven fibers. Here we have two scaffolds made from the same material but with different stiffness and pore sizes. The cells shown in orange interact with the cell scaffold, which is in blue or teal. Now, if we look closely, we can see that the scaffold on the right has more cell growth and migration. So why is that? Well, the scaffold on the left is stiffer and has larger pores, making it harder for the cells to spread compared to the more compliant scaffold with smaller pores on the right. And this demonstrates how both architecture and the mechanical properties of the scaffolds play a crucial role in guiding the cell behavior. We can also design spatially varying 3D architectures incorporating regions with high stiffness on one side and lower stiffness on the other. Additionally, we can introduce areas with enhanced stretchability, enabling tailored mechanical responses within a single structure. And by doing so, we can also program the deformation behavior of these 3D microarchitectures. For instance, upon stretching, certain regions deform more than others, revealing the emergence of a pattern that we are pretty familiar with. And in case this is not obvious, it is actually our MIT logo. So to summarize, we demonstrated a framework to locally tune porosity, stiffness, and the deformation behavior of 3D bioscaffolds in three-dimensional space without needing to change the material composition. This not only allows us to study, but also control cell behavior by designing the ideal microenvironment for each cell type. So we'd like to thank the Portela Research Group, Molly Carton, and the Zhao Lab for making this research possible. Thank you for listening.